The blood is a tissue builder mainly by two cells, the red globes and the white globes. The red globes are in charge of distributing the oxygen in all the body, but the mature red globes lose their nucleus, but them are still being eukaryotic cells, even if they already lost their nucleus. By the other hand, the white blood cells are the ones in charge of the defenses of the organism against foreign substances or infectious agents. So we can say that the white blood are like an immune system for each cell. Also, the white cells are the ones who carry the DNA information. So they are like the ones who carry all our genetic information that is inside of our body. And they also have a defined nucleus. By defined nucleus, we can say that the nucleus is correctly limited and organized. So we have like a membrane that has their chromosomes delimited, so they are like pretty organized. The objective for this laboratory is to isolate the DNA from a blood sample. By this, we say that we like separate the red blood cells from the white blood cells, because the white blood cells carry the information. So we conclude that they carry the DNA. So they are the ones which we are focusing on and we are interested on. For doing this, we made several chemical procedures that are later going to be explained and used. By the other hand, according to Mao and Jie in 2012, she said that with DNA extraction we can make many applications for this process, and they make like our daily life easier. These applications are
pharmaceuticals. DNA extraction is used as the initial step in manufacturing a number of pharmaceuticals. Pharmaceuticals made via recombinant genetics include bovine growth hormone, the BVH, and human growth hormone, the HGH, in addition to a number of other hormones. One of the most used is called the insulin. Medical diagnosis. Diagnosis of certain medical conditions can often be made from DNA extracted from a patient. Conditions that can be diagnosed by genetic testing include cystic fibrosis, sickle cell anemia, Fragile X syndrome, Huntington's disease, hemophilia A, Down syndrome, and Tay-Sachs disease. In addition to diagnosing existing diseases, also common is testing to see if a person is carrier of a particular disease but does not have the disease. Identify verification. A well-known use for genetic extraction is genetic fingerprinting, a process in which genetic material can be matched to genetic material at crime scene. For example, genetic verification has worked both to place a person at the scene of a crime and to exonerate people falsely convicted of a crime. We can observe this case of use of DNA in a TV series called Criminal Minds, in which they try to match DNA samples to find a crime responsible. We took the blood sample and applied it into 11 milliliters of buffer TE to reach into the test tube at a volume of 13 milliliters. Then we shake softly and then we centrifuge the sample at 4,000 RPM during 10 minutes. After that, we took off the test tube from the centrifuge and we add 1 milliliter of Lysis buffer and we apply it into a vortex from 1 minute. Later on, we add 67 ULD of CDS and we apply it 1 minute into a vortex. Then we add a 10 UL of proteinase K and after we shake softly, we add 335 UL of NaCl and we apply it into a vortex from 1 minute. After that, we centrifuge the sample at 4,000 RPM during 15 minutes again, and after we took off the sample, we transferred the Super9 into a test tube. Later on, we add 2 volumes of ethanol and we centrifuge again the sample during 10 minutes. Finally, we took off the sample of the centrifuge and we start to see the DNA.
The laboratory practice objectives were accomplished. The DNA isolation was successfully achieved because it remains alone, as you can see in the image, without membranes or other stuff that may contaminate the sample. We conclude that this is a molecular biology technique because with this we can isolate DNA from blood sample, and also with this kind of experiment is useful to do paternity tests, clinical diagnosis and scientific investigations.